welcome to the Ather Talk podcast, the series hosted by the EAS Young Fellows. My name is Azin, Kheirkhah, and I'm from the Institute of Genetic Epidemiology at the Medical University of Innsbruck. Today, we're thrilled to continue the series of interviews with Professor Paul Ridker at the 20th International Symposium on Atherosclerosis in Muscat, Oman. Professor Ridker is a Eugene Braunwald Professor of Medicine at Harvard Medical School and the Director of the Center for Cardiovascular Disease Prevention at Brigham and Women's Hospital in Boston. Remarkably, Professor Ridker has authored over 500 peer-reviewed publications and led numerous clinical trials, including the Jupiter, Cantus, and Prominence studies, which have significantly influenced the understanding and management of atherosclerosis and cardiovascular risk. His work on the C-reactive protein as a biomarker has reshaped cardiovascular prevention approaches. On behalf of the EAS Young Fellows, I would like to thank you, Professor Ridker, for accepting our invitation.
Azin, it's wonderful to be here. Thank you for having me in Oman, and I very much look forward to the EAS meeting in Glasgow.
Thanks a lot. So first off, I would actually like to congratulate you on being awarded the Bartušková Prize. This is a prestigious award, and I actually wanted to know what do you think and what do you feel personally for being awarded the Bartušková Prize, and does it influence your professional journey, and how does it influence you personally as well?
Well, it's a great honor whenever one is recognized by one's peers for work that one has done. But the reality is, I think that most of us get the real rewards from the research we do and from the sense that perhaps what we do will help someone's health somewhere on the globe. The awards are certainly a nice recognition, but at the end of the day, what sustains a long career in biomedical research is the belief that you're actually going to change medical practice.
Yes, that's the importance, exactly. So, Professor Ridker, in our interviews, so we have a lot of early career scientists who are listening to this podcast, and one thing we want our listeners to hear is that who is the person behind this great science. So we will ask you a little bit of personal questions, and the very first one is, how does a typical day in your life looks like? So do you even have a typical day, or can you elaborate on that for us?
Well, one of the exciting things about living a very academic biomedical life is, in fact, that my days are quite different. I like that. But a typical day, I am up very, very early, don't sleep very much. I will take my golden retriever for a long walk and take a few phone calls. Usually I'll work out for about an hour in the morning. Fitness is very important for lots of reasons over life. I've played competitive tennis my entire life. When I was younger, I was a good tennis player. Now I just attempt not to get injured is my main goal. And then on a typical day, I might go to the hospital if I had clinical duties. I might go to my research lab. In this era of post-COVID, it seems like we have a lot of meetings still on Zoom, even though I'd rather they were face-to-face. By the way, that's a very important issue for early career individuals. The time you have face-to-face with your mentor and with your other colleagues is very important. It's relatively easy for a senior person to perhaps live their life on Zoom. It's not so good a way for young people. So I encourage people to go into the office, be with people, what we call water cooler time. It's very important to understand in terms of how hypotheses are generated, to learn scientific techniques, to see what's exciting and going on. But my day varies a great deal, and I actually like that about my life.
Yeah, that's really interesting to hear, actually. And so you talked about tennis. So I guess that's one of your hobbies outside your work. But I mean, do you have other hobbies as well, or interests outside your work?
Well, my wife would say she's a tennis widow because many years ago, when we were thinking about how, as a preventive cardiologist, could we impact care not only through our research on a global basis, but in our own backyard on a local basis, we established a health and wellness center at an inner city tennis center in Boston. So this is a very unusual tennis center because it's in a neighborhood of very socially deprived, very economically deprived area of Boston.
Yeah.
Where we now have after-school education programs. We have a health and wellness program. but it is situated inside of a tennis center, and I happen to be the board chair of this organization. It's called Sportsman's Tennis and Enrichment Center. We made a large donation with some of our patent money many years ago to establish this health and wellness center. And it gives me a good feeling about what we can do locally, as well as what we might be able to do nationally and globally. That's really impressive. And it's really nice to hear that a great scientist like you actually also thinks about the local as well. So also another question is how important is this work-life balance to you, and how much of it do you really apply in your life? Well, that is a complicated question. I have done some things in my professional career that were very purposeful to make my work-life balance easier. Perhaps the most important is that for personal reasons as well as professional reasons, I have not taken on any major administrative jobs. So I have never served as chairman of medicine or the head of the hospital or in any of those kinds of positions. Now, my colleagues who do that, I'm very impressed. And I'm very glad they do it, particularly if you have very good leadership. It makes running the rest of our lives terrific. But you have to look at yourself in the mirror and say, what are the things that make you happy? What do you think your own skills are? And while I am confident I probably could be a good administrator, I don't think that would make me the happiest. I'm happier when I'm doing my research. I'm happier when I'm seeing my patients. I'm happier when I'm at home. And so that was a very conscious choice to say, these are the things that will keep me sustained. And I'll let other people run the hospital and run the medical school. That's great to hear. So the happiness is more important rather than the position or the hierarchy or like getting these administrative positions. Again, I think you have to do what you feel best doing. Yes. There are people who are extremely talented at administration, just as there are people who are extremely talented at clinical care, just as there are people who are extremely talented at research. You're lucky to be good at any one of these things. Very few people are good at all of them. Yeah, exactly. Thanks a lot. So you trained in medicine, and but what did you always? But did you always have a curiosity to do research, or was it something that developed during your training, or is it a difficult question? It's not a difficult question, but life is not so linear. In fact, I thought I would be a surgeon for a long time. I like doing things with my hands. My other passion in life is construction and woodworking. But when I did get to medical school, I found internal medicine to be much more intellectually challenging and much more interesting. I had, when I was younger, worked at the National Institutes of Health in Bethesda, Maryland, in basic immunology research lab. So I also thought about being a basic scientist for a long time. And then later on, when I was in medical school, I took an extra year and I lived in Zimbabwe for a year. I was interested in parasitology and infectious disease. This happened to be just at the beginning of the AIDS epidemic. And so the experience was quite challenging in terms of the patients we saw. But I think my long-term interests in inflammation biology, innate immunity, and atherosclerosis come out of these early experiences where I was studying in an immunology laboratory at one point in my life, and also these life experiences in Zimbabwe and South Africa where you saw a immunologic disease taking such a toll. But I don't think you can plan these things. I think they just happen in your life. Yeah, I think you're already answering my next question is like, how can young researchers find their niche as well? So like your focus was on inflammation and cardiovascular research. So if someone like us, the junior scientists, how can we find our niche? I mean, what are the criteria that you think are very important to find one's niche in the research field? Or as you said, it's not very easy to say how you can find it, right? Well, again, so far the story has been about my intellectual interests and my sort of social interests that had to do with inflammation. I never thought I was going to be a cardiologist. It wasn't even on my list of possibilities until I did my residency. So when I came to the Brigham and Women's Hospital in Boston, the chief of cardiology and chief of medicine was Eugene Brownwald. And back then, our house staff was a very small program. and we worked extremely hard because we were on call pretty much every other night, and it was a very busy time. At the end of the year, Professor Brownwald meets with each of the young interns, and at the end of my discussion with Dr. Brownwald, he said, "What are you interested in doing?" And I mentioned to him my interest, and he said, "Well, I think you should be a cardiologist instead." And I said, "Okay." And that's really what happened. Yeah. Because he had such a huge influence on cardiovascular medicine, and he's such an influential individual. One of the best decisions I ever made.
So a great mentor is one of the key factors for finding
Absolutely.
a niche,
I think that young people need to seek out individuals who are senior to them, who can actually sit down with them and talk about what's best for them moving forward. It may not be what was best for the mentor. The more senior person, he or she might have succeeded in one direction, but the younger person would succeed in a very different direction. I think the most important things in a academic career are to find, again, two things work. Try to figure out what you're actually very good at, and then try to find a problem that excites you. I don't recommend attempting to do something you're not good at. And I see this a lot. People say, "I want to be a computational biologist," but math is not their strong point. Someone else who does have a particular skill at the bench or with their hands or with the way they see things, that's the way to go. It's much easier to be good at something you're actually naturally good at. And then it's important to find a question. It's possible to have a successful career where you change the question quite often. Many people do that. In my case, for all practical purposes, I've been asking the same question for almost 35 years now. And that made it difficult, but it also meant that I could focus on a series of ideas and a series of questions that would take different skill sets over those years to figure out. And that's very exciting when you get to be mid-career and change your skill set. We didn't change the question we were asking, but we changed the skill set. So while I'm trained classically in population biology and epidemiology, and that's where much of the inflammation work first came from, it soon became apparent that to actually change clinical practice, that would never be enough. And so we had to learn how to do clinical trials because that would change guidelines and change practice. But another part of our research had to do with the fundamental determinants of inflammation, and that required me to go back and train in genetics. You're a geneticist. Yeah. But in my day, you had to actually retrain. We didn't take genetics courses in school. Yeah. And that was very exciting to realize in mid-career to sit down and learn some genetics. Yeah. And within a few years, we were running some of the largest, earliest genome-wide association studies at a time when other people hadn't started doing this yet. And also I have the opportunity to interact with tremendously bright young people on a regular basis. One of the wonderful advantages of being at the Harvard Medical School is we get brilliant young people coming in every day. And part of our job is to inspire them to be interested in what we do. And very often, the way my research lab works, there are some people whose skill sets are similar to mine, but many people whose skill sets are extremely different from mine. A computational biologist, a statistician, someone who really thinks about chemistry in a more sophisticated way than I might. These people are fun to work with, and they really expand the directions we can go and have been invaluable in trying to understand ultimately how to bring this information to patient care.
That's amazing because these are the collaborations that further the research, actually the progress of the research, and it's very important. So Professor Ridker, your work has successfully translated the importance of inflammation in cardiovascular disease from bench to bedside. So can you elaborate for us, how did you bridge this gap between research and its use in patient care? And what were the obstacles you faced and how did you actually overcome them?
I think at the end of the day, I'm a physician. You look in the mirror and you say, what is it that you see yourself as? So I think I always knew that it was important to bring this back to the patient and back to the bedside one way or the other. Population biology is complicated because You can make observations about groups of individuals. Mm-hmm. Many of our studies have tens of thousands, if not much larger numbers of patients in them. But you try to figure out, how can I then take this back to the individual person I might see in clinic the next day? That's a big challenge. But I think it also makes this exciting. And I wish I could tell you there was a grand scheme and a grand plan here, but that's not the truth. Things changed as we went along and as we began to figure things out.
Thanks a lot. So, and you're also one of the most cited researchers in the field. So have you observed a substantial shift in the publication dynamics during your career?
I think there is more pressure on young people to publish in terms of volume than there used to be. I don't think that's a good thing. I think all researchers at all ages should really concentrate on quality over quantity, if at all possible. If you ask the right question and you develop the right protocol and you think through the ideas, in most fields, whether it's bent science or clinical medicine or even translational medicine, if the experiment is done correctly, whether it's neutral or positive, it will get published in a good journal either way. It's the poorly designed experiment that will not find a good home. But we have so many journals today and so many other avenues of getting information out that it can be very difficult to get other people interested in what you're doing. Again, in my particular field, many of our projects take 6, 7, 8 years to complete. It can be very disappointing when they don't work. It can be very exciting when they do work. But the biology is what has to sustain it.
Exactly. And I think the problem that young scientists face right now is that they are being assessed by the number of publications they have. And that's why we have to keep on publishing whatever results we have. And that's, as you said, there's an overload of information and probably the quality is not as good. And do you have any tips on how we can filter out, how do you manage this, when you're looking for an article for an answer to a question you have, how do you filter out good papers from this bunch of overload information?
Well, there's two different sides of this. The first is, there's a reason we have very high quality journals, and those should be the journals that you aspire to publish in, and those are probably also the journals that you should spend most of your time reading. So if it's a basic science world, it's either Cell or Nature or those kinds of journals. If it's a clinical world, it's going to be the New England Journal or the Lancet, things like that. And in the cardiovascular world, it's very clear. But I think you really should trust the editors, trust your mentors. In your own specific area, there'll also very likely be editors and people who respect exactly what you're doing. Remember, you're not trying to influence huge numbers of readers. You're trying to influence the small number of people who actually are very interested in what you're doing. So I like the poster sessions at most meetings, because a young person will be standing there with their poster, very proud, and then a major player, a major person in the field comes up and starts asking them direct questions. They're there because they're really interested in what you're doing. So that's very good. I would say, though, there's two other things I recommend to all the young people I do talk to, and think about this. And there are two things that give them some anxiety. So I just warn you up front. One of the most important things that any successful scientist will tell you, if they're honest, is the huge role that luck plays in a scientific career. I highly recommend being lucky. What do I mean by that? Many scientists and many investigators work very, very hard, but the hypothesis they were chasing, even though it made complete sense, just turned out not to be the way nature works. And there's nothing you can do about that. On the other hand, someone may be doing an experiment and it exactly is what nature actually does, and they're suddenly very successful. I think both of those people should recognize one was just lucky and one was unlucky, that maybe it wasn't how hard you worked or how brilliant you were, it just had to do with luck. And I think that's a very important thing that everybody should remember in the biologic investigation. Now, there's a second piece of advice I give young people, which also causes some anxiety, and that is be willing to change direction. You can work on an experiment at the bench or in a clinical setting for many years, and it may not seem like it's going anywhere. There's two possibilities. One is you're almost there and you haven't gotten there yet, but the other is maybe it's not going anywhere. And sometimes it's difficult to say, this particular piece of research is not heading where I either had hoped it would head, or the information I'm getting back from my experiments or my data don't really support the hypothesis I thought I had. It takes a great deal of courage to say, perhaps this is the wrong direction and they need to change direction. And again, we were both lucky and we changed directions many times. And I think these are qualities of tenacity and persistence that really matter in the long run.
Exactly. Thanks a lot for these two important advices. So clinical trials are an integral part of your career. You have designed, conducted, and served as a trial chairman or co-chairman in trials substantially advancing the treatment of cardiovascular disease. What would you say is the most challenging part of a trial design? And what is the major reward you get from it?
We began to do clinical trials because it was very important to actually change clinical care and try to shift the way guidelines work. And at the end of the day, that's going to be driven by randomized, double-blind clinical trials. So this was a skill set that we had to learn. In my case, it's a little different than other clinical trial research groups. There are excellent, superb clinical trial research groups all over the globe, many of whom focus entirely on the performance and conduct of clinical trials. And they do very good work. But ultimately, they're doing different kinds of trials in different settings. That's not what I wanted to do. I wanted to use clinical trials as a way of advancing the ideas that we were interested in about inflammation and biology. So I have spent much of my time trying to convince either the National Institute of Health, our federal funder, or various pharmaceutical companies to do the clinical trials that we wanted to do that were very specific to the questions we were asking. So it's a little different than clinical trial groups that are doing superb work, but are often running clinical trials as their primary aim. Our aim was the biology, and the clinical trials became the method of getting there. That being said, running clinical trials is a lot of fun. First, you get to work with hundreds, if sometimes thousands of individuals, many of whom become lifelong friends and collaborators. And in the clinical trials that we've done, I have many times the same investigators year in and year out. And that's it becomes a big family. And particularly for people who perhaps are living a life that's clinically active, academically active, but may not be lucky enough to live in London or Boston, or it's a great way to be involved. So I have very close friends all over the globe, which makes my life a lot of fun. Get to travel to see friends everywhere we go who have been participating in clinical trials with us. And we have to remember that without the physician leadership, without the coordinator leadership at every single site, none of this would ever happen. So it's actually a pleasure and a very nice way to see how this can work.
Yeah. Thanks. And is it rare or is it common to encounter unexpected results during clinical trials?
Well, learning how to handle results that you did not think you were going to get, as I said before, is a very important issue in terms of changing direction. It's also a very important issue in clinical trials. Clinical trials give you a truth. They give you one truth, not the only truth, but they do give you one truth in a general, large population setting. Will this intervention improve outcomes or not? We don't talk about our studies that we've done that were neutral on a regular basis. But it's not just the pemafibrate study that did not work out for us initially. We did another inflammation reduction trial using methotrexate that did not work. I actually ran some PCSK9 studies where the monoclonal antibody that we used ended up having some antibodies were developed, and it just didn't work in a commercial space. So you learn from these experiences and hopefully don't make the same mistakes a second time.
Thank you so much. And looking ahead, what do you think is the next frontier in inflammation-focused cardiovascular research?
Well, at the EAS meeting in Glasgow and also here in Oman, we're describing several clinical trials we're doing with interleukin-6 inhibition. So remember our CANTOS trial. This was using a monoclonal antibody called canakinumab, which is a fully human monoclonal that targets interleukin-1 beta. That showed a 15 to 17% reduction in heart attack or stroke without any change in ApoB or LDL cholesterol. So from a historic perspective, that clinical trial is the proof of principle that targeting innate immunity can actually lower cardiovascular event rates just as much as giving lipid-lowering drugs. That's very exciting. But the question is, where in this particular cascade between the NLRP3 inflammasome, IL-1, IL-6, ultimately down to our biomarker C-reactive protein, where do we want to target this therapy? For a variety of reasons I don't want to go through today because it's just sort of a little bit of dull science, I suppose. We think that that target might well be interleukin-6 directly. And so again, working with industry colleagues, we helped to develop a novel IL-6 ligand inhibitor, a drug called ziltivekimab, and we completed some phase 2 data showing that this does lower the target we want. And working now with Novo Nordisk on a global basis, we have multiple ongoing large-scale outcome trials looking at IL-6 inhibition, first in the setting of chronic kidney disease and atherosclerosis, second in heart failure with preserved ejection fraction, and thirdly in acute coronary ischemia. All three of these settings have a very profound pro-inflammatory milieu, and the question will be, can we, in addition to lipid lowering, in addition to ACE and ARB, in addition to GLP-1s, can we actually target the inflammation itself, and will that safely deliver benefit for our patients? These are unknown questions, and this is what makes a scientific life exciting.
Thanks a lot. And do you think there are any overlooked topics in atherosclerosis or cardiovascular research in general, as in besides lowering lipids and also targeting the inflammation, do you have some secret hint to which other pathways may be of great importance in the future?
Well, this is what makes science so interesting.
Exactly.
Let me give you an anecdote. I'll go back to Professor Braunwald again because he has been the guiding light for most of my career. When I was in training, my copy of Heart Disease, the Bible of cardiovascular medicine, didn't have a single chapter in it about lipid lowering or statin therapy. Now think about that, right? This is the bedrock of our therapy today. And I'm not that old. So it's very important to recognize that the textbook is going to change, and it's going to change many times during your scientific lifetime. And you're going to add to those changes. I think about the amazing things we're learning today from proteomics, from genetics, that you're in field that you're in, to areas like clonal hematopoiesis. We didn't even know it existed 8 years ago, and now we have huge development programs targeting this. So I think the biology will always find new ways to stimulate us. I cannot imagine we'll be treating patients the same way we do today 30 years from now, just like we don't treat them the way we did 30 years ago. Clinical care moves forward slowly, but it moves forward because of young people learning new things.
Thanks a lot. So to actually finalize this interview, I have one last question. So in these series of interviews, we have a section which we actually call it the graveyard of ideas. So it's a section for the failures the great scientists like you have faced. So because we always hear all success stories, and there's not so much about how many failures did people have. And I think this is important to give a motivation to young scientists that you are not alone. If you do face a failure, what do we have to do? And do you have a personal story that you would like to share with us?
Yeah, it's a great question. And it's a very important issue to have the resilience to bounce back from data that doesn't make sense. And we already talked a little bit about having to change direction. Let me tell you about, so it's kind of funny. One of the magical things that sometimes happens is when you can take what's probably your greatest failure and try to turn it into something positive. This is like magical thinking a little bit, but I'll give you the example. When we were trying to definitively test whether or not inflammation inhibition per se reduced cardiovascular event rates, it was around 2008, 2009. We had completed a clinical trial called Jupiter. This was a clinical trial that said statin therapy, which we had shown 15 years earlier had anti-inflammatory benefits. We had shown that statin therapy could lower cardiovascular event rates enormously in primary prevention, even when LDL cholesterol levels were low. But that was not a test of the inflammation hypothesis of atherosclerosis because we used a drug that also lowers cholesterol. Now, when I speak publicly, I normally talk about CANTOS because it's a very positive study. The truth is, at the time, we designed two parallel studies. One was CANTOS that you do know about. The other was a federally funded clinical trial called CIRT, C-I-R-T, the Cardiovascular Inflammation Reduction Trial, where we tested low-dose methotrexate. Now, at the time, I thought each of these had an equal chance of success and an equal chance of failure. Fast forward 7 years, enormous amounts of work on both studies. One is a tremendous success. One is a total failure. But both ended up in the New England Journal of Medicine. And that's part of the lesson here, is that if it's designed, the failure of methotrexate, which obviously is a very good drug to treat rheumatoid arthritis, the fact that it did not lower IL-1, that it did not lower IL-6, it did not lower CRP in the setting of atherosclerosis was something very important for us to learn. Whereas canakinumab, in the exact same setting, did all those things. So the null or neutral data from the methotrexate trial, which I honestly thought would be just as likely to work as the other, taught us that maybe the pathway by which you reduce inflammation is very, very important. But we did not know that going in. And the amount of work that goes into these, nobody does a multinational, large-scale randomized trial hoping for a null result. The question is, what do you do with that information and how do you bounce back from it and keep moving forward?
Thank you so much. This was very insightful. That was an incredible interview with you. Thank you, Professor Ridker, for sharing your time and insights with us. Also, a big thank you to all our listeners. This podcast is for you, and we need your feedback to tailor it to your interests. So please don't forget to leave us your impressions or suggestions that you would like to hear more about. Again, thank you so much, Professor Ridker. And we are looking forward to your Anishkov lecture in Glasgow 2025 EAS Congress. And this was a great honor for us to have you here and get your insights and advice.
Azin, the honor is mine. It's wonderful to see young people like yourself pursuing scientific careers. I look forward to seeing you and your early career investigators in Glasgow next year.
Thank you very much.